Nguyen, and thanks for joining me as we learn about the structure of the cell. By the end of this lesson, you will be able to do three things. This lesson consists of 4 sections. Note that at the bottom left-hand corner of each slide is an indication of which section the slide belongs to. This way, you can easily find where you are in this lesson. So what are membranes and why are they important? Let's start with a question. Have you ever taken a spoon of salt or sugar and tossed it into a swimming pool? What happens to the salt? It dissolves, right? But have you ever jumped into a swimming pool, went for a swim? Did you dissolve? Why not? Well, the answer is membranes. A membrane is essentially an oily layer. You can think of a membrane as a peanut butter sandwich. The outer 2 layers of the sandwich is bread. Bread mixes with water, it likes to soak up the water. But the middle layer is peanut butter. It's oily, it's fatty, it doesn't mix with water. So the oiliness of a membrane prevents water from going through it. Another cool feature about membranes is that they can bend. They can bend and fuse and butt off to form hollow spheres. So the ability of membranes to block the flow of water, to bend, and to pinch off forming hollow spheres allows cells to use membranes to build incredible structures inside of those cells. Let's now talk about the structure of an animal cell. To understand the inner workings of a cell, it's helpful to think of the cell as a city. Just as a city has institutions that make sure there is law and order, electricity, clean water, sewage disposal, and efficient transportation, the inside of a cell has similar structures that each does its own part to make sure that the cell is running well. Cities have City Hall, where the mayor, right, and the court system is often located. Right, cities have manufacturing companies that produce, cement and lumber and building materials, cars, tractors, trucks, so on and so forth. Cities also have a post office to deliver, mail to the right places. Cities have power companies that produce electricity. Cities have waste disposal facilities that send trucks to pick up your trash. Cities also have a sewage disposal facility, right, which removes your sewage waste and recycles it, right, or filters it. Cities have recycling centers, right, to make efficient use of raw materials. And lastly, cities have schools that make sure students are educated as they prepare to enter adulthood. As it turns out, the inside of a cell contains distinct structures called organelles. These organelles each serve a function that is very similar to the various facilities that maintain the function of a city. For example, in the cell, City Hall is the nucleus. Manufacturing companies that produce raw materials is like the smooth endoplasmic reticulum and the rough endoplasmic reticulum inside the cell. The post office of a city is similar to the Golgi apparatus inside the cell. The electricity company in the city is similar to the mitochondria inside a cell. The waste disposal facility is similar to what's called the lysosome inside the cell, and the sewage disposal company is similar to what's called the peroxisome inside the cell. The recycling center of a city is similar to the vacuole that is inside of the cell, and the schools inside of a city are similar to what's called the ribosomes inside of a cell. Here is a diagram of what inside of an animal cell looks like. It's important to study this diagram and be familiar with the relative shapes and locations of these various organelles. The nucleus contains DNA, which is packed in the form of chromosomes. The smooth endoplasmic reticulum, or SER, makes cholesterol. It also has a job of detoxifying toxins, which are chemicals inside the cell. The rough endoplasmic reticulum, otherwise known as the RER, has ribosomes on its surface, thus it looks bumpy and is called rough. The RER makes proteins that will be shipped in vesicles, also known as membrane pouches. These vesicles are shipped throughout the cell. The Golgi apparatus, also known as the Golgi body, receives pouches of protein that come from the rough ER. The Golgi then adds address labels on these proteins, and then the Golgi ships these proteins to different parts of the cell and even outside of the cell in the form of vesicles. Again, vesicles are membrane pouches. The mitochondria looks like this. Its inner membrane is folded. The mitochondria produces energy molecules for the cell and is known as the electricity company or the power company of the cell. Lysosomes break down unwanted organelles, old organelles, damaged organelles, or just unwanted molecules in the cell. The peroxisome gets rid of harmful chemicals called peroxides. Peroxides are byproducts of the normal chemical reactions that happen inside of the cell. The vacuole, which is only in plants, is special in that it stores water and chemicals for later use. The vacuole is responsible for taking in a lot of water and causing the plant cell to swell and enlarge. Ribosomes make new proteins. Ribosomes can be free floating inside the cell, but outside of any other organelle. And ribosomes can be attached to the surface of the rough ER. There's another structure inside the cell that's important to know, though it's not an organelle because there are no membranes around it. It is the centriole. Centrioles are diagrammed here, and here's an enlarged image of them. Centrioles occur in pairs, and the pair is called a centrosome. The centrosome and the centrioles inside them serve as the anchor for a dividing cell to pull itself apart. When a cell divides, there are two centrosomes, one on each side, and these are the anchors from which the railways that push and pull the chromosomes extend. Animal cells can have tails that come in two forms, cilia and flagella. The tails are for paddling liquid, swimming, or sensing chemicals in the liquid environment around the cell. Flagella, or one flagellum, moves like the outboard motor on a motorboat. It spins, pushing water. Cilia, on the other hand, move like the oars of a rowboat, paddling back and forth to move water. In humans, the cells that line our airways have a lot of cilia. The cilia are constantly beating back and forth to move mucus inside our lungs up to our throats. In humans, sperm cells have flagella that's help the sperm swim up the reproductive tract of the female in search of the egg. Let's now talk about the structure of plant cells. Plant cells are similar to animal cells, except for three key features. They are the chloroplast, the vacuole, and this thing called the cell wall, which in this diagram is the thick wall in color green outside of the yellow, which is the cell membrane. Chloroplasts are the organelles that do photosynthesis to produce sugar for the plant. Chloroplasts are the reason why plants have the color green, and actually the color red and yellow. The vacuole is a large organelle that stores water and chemicals inside of it for later use. Vacuole is a membrane pouch that swells with water to create tension inside of a cell, as if the vacuole were a water balloon. The cell wall is a firm wall of fibrous material that is outside of the cell membrane. It gives a rigid structure to plant cells. The cell wall is primarily made of a polymer called cellulose. Here's a diagram showing the similarities and differences between an animal cell and a plant cell. For your convenience, here's a table that shows the key differences between an animal cell and a plant cell. You should know these differences. Let's now talk about the structure of bacterial cells. Let's first define some important terms. Animal cells and plant cells are known as eukaryotic cells. They are eukaryotes. A bacterial cell is a prokaryote. The word eukaryote means true nucleus. Eukaryotic cells have organelles that are completely bound by membranes, such as we discussed with animal cells and plant cells. Prokaryotes, however, do not have membrane-bound organelles inside of them, though they do have infoldings called inclusions, which we'll talk about. But the difference between a eukaryote and a prokaryote is a very important distinction to know. All living things fall within one of three domains of life: eukarya, bacteria, and archaea. The main difference between eukarya, bacteria, and archaea is that eukarya have membrane-bound organelles inside of them. Bacteria and archaea are both single-cell organisms, and the difference between bacteria and archaea is in the structure of their cell wall and also their method of getting food through various metabolic pathways that are distinct. Bacterial cells are a lot smaller than animal cells or plant cells. They are much similar in structure, but that doesn't mean they're less complex or easier to understand for a biochemist or a microbiologist. Bacteria do not have membrane-bound organelles inside of them, so everything inside is free-floating, or most of the things are free-floating. Instead of a nucleus, the bacteria have what's called a nucleoid, which is a ball of yarn that is the DNA. Bacterial cells have a cell membrane that encases everything inside of it. They also have a cell wall outside the cell membrane and a protein capsule outside of the cell wall that is sometimes part of the cell wall. Bacteria can have tails in the form of flagella that spin and help the bacteria to swim. Bacteria also have projections called pili. One projection is called a pilus, but many projections are called pili. Pili are very useful for communicating with other cells. Pili are like tubes through which two bacteria can connect their insides to each other, such that one bacteria can send DNA and other molecules through that tube to a neighboring bacteria. Though bacteria do not have membrane-bound organelles inside of them, they do have something close, known as inclusions. Inclusions are infoldings of the plasma membrane. The advantage of having inclusions is that you can store polymers there. You can prevent the increase of the saltiness of the liquid inside of the bacteria if you store sort of salty molecules in these little membrane infoldings. These infoldings, just like regular organelles, can house specific chemicals that the bacteria wants to isolate until the right time when it needs to do certain reactions. One type of inclusion inside bacteria are known as magnetosomes, for magnetotaxis, which means moving in the direction of a magnetic field. Believe it or not, by storing a series of pouches that contain iron atoms inside of them, a bacterial cell can sense the north, south, east, and west directions of the Earth, and thus know where to swim in order to get to where it wants to go. You heard me right. These ancient bacteria developed cell phone technology long before humans invented it. Another example of a bacterial inclusion are chromatophores. Chromatophores are enfoldings that contain the enzymes necessary for the bacteria to do photosynthesis, that is, to use, uh, say, light energy and carbon dioxide gas to produce sugars. This is the end of our lesson. Thanks for joining me as we learn about the structure of the cell. This knowledge is fundamental to so much of medical practice and medical advancement. I hope to see you next time in other lessons.